Ferroptosis is a form of programmed cell death, one of the mechanisms that causes cells to die. It is characterized by the accumulation of damaged lipids as a result of increased reactive oxygen species dependent on intracellular ion metabolism, which ultimately leads to cell death through the destruction of cell membranes and intracellular organelles. In vivo, ferroptosis has been shown to suppress cancer. In addition, a number of studies are underway to induce ferroptosis in cancer cells and use it to treat cancer. However, it is not clear what other significance ferroptosis may have. Dr. Hironari Nishizawa and his colleagues at the Department of Biochemistry, Tohoku University Graduate School of Medicine, have discovered that ferroptosis is involved in the suppression of cellular and organismal aging. The research group previously found a link between the transcription factor BACH1 and ferroptosis. Transcription factors are proteins that promote or repress the function of various genes, and BACH1 usually represses its target genes. The genes repressed by BACH1 included those that prevent ferroptosis. Suppressing genes that prevent ferroptosis means that BACH1 acts to promote ferroptosis. The group also found that producing large amounts of the BACH1 protein can drive cells into ferroptosis without the use of drugs. The study found that when this happens, the cells release substances that inhibit aging. Ferroptosis was induced in cultured cells and the culture supernatant was collected. When administered to another cell, it inhibited cellular aging. These pictures visualize the amount of the enzyme S-beta-galactosidase. S-beta-galactosidase is an enzyme that increases as cells age, and its quantity can be used to determine the extent of cellular aging. In cells treated with ferroptotic culture supernatant, the blue color is lighter than in controls, indicating a lower amount of S-beta-galactosidase. It means that cells treated with the ferroptotic culture supernatant suppressed aging. Other examinations comparing cell proliferation rates, DNA damage repairability, and expression of senescence characteristic genes also indicated that ferroptotic cell supernatants inhibit cellular aging. A possibility emerging from these results is that cells undergoing ferroptosis released some substance, which had the effect of inhibiting the aging of other cells. The question is, what the substance is. The researchers found that the cell growth factor FGF21 was increased in BACH1 overexpression cells. Growth factors are small proteins secreted in the body, and exert a wide range of effects, such as promoting cell growth and differentiation, and altering metabolism. Among these, FGF21 has been attracting attention for its ability to improve lifestyle-related diseases, such as obesity and diabetes. Examinations found that BACH1 is able to increase the amount of FGF21 in the cells. So, the following hypothesis was developed. Overproduction of the BACH1 protein leads to FGF21 increase. At the same time, large amounts of BACH1 cause ferroptosis. At this time, FGF21 is released, which exerts an anti-aging effect on cells. To test this, BACH1 was overexpressed in two types of cells to drive them into ferroptosis: normal cells and cells lacking the gene that produces the FGF21 protein. If the anti-aging effect of the ferroptotic culture supernatant was due to FGF21 released at this time, the supernatant of FGF21 deficient cells, in which the FGF21 protein would be absent, should fail to show the anti-aging effect. Administering culture supernatants from normal ferroptotic cell culture to other cells showed an inhibitory effect on aging in the administered cells. On the other hand, as expected, the ferroptotic supernatant of FGF21 deficient cells did not show the anti-aging effect. Therefore, the anti-aging substance in the ferroptotic supernatant was shown to be FGF21. We now know that BACH1 induces cellular ferroptosis and increases the amount of FGF21 in cells. The FGF21 that is released outside the ferroptotic cells inhibits the aging process in other cells. Does the release of FGF21 during the induction of ferroptosis by BACH1 have health implications? The researchers investigated the relationship between induction of ferroptosis by BACH1 and obesity. Mice are fed a high-fat diet and weighed. Body weight was compared between wild-type and BACH1 gene-deficient mice. BACH1 deficient mice lack the BACH1 transcription factor in every cell. The absence of BACH1, which is involved in the induction of ferroptosis, makes them less likely to develop ferroptosis. Weight gain due to a high-fat diet was more pronounced in BACH1 deficient mice. Continued high-fat diets often leads to fat accumulation in the liver and the development of fatty hepatitis. At this time, cell death by ferroptosis was found to be occurring in the liver. FGF21 levels in the blood of mice fed a high-fat diet were lower in BACH1 deficient mice than in wild-type mice. Low levels of obesity-suppressing FGF21 may explain why BACH1 deficient mice are more likely to be overweight. This means that in wild-type mice, conversely, induction of ferroptosis by BACH1 increases FGF21 in the blood and suppresses obesity. In addition, the effects on the lifespan were investigated. These curves show daily survival of mouse populations. The blue curve shows that almost all mice in this population survive the first 50 days. Thereafter, the number of survivors gradually declined, reaching half in about 90 days. And finally, around 170 days later, the last survivor died. So, if the curve is located to the right, it means that the population had a longer lifespan. The blue curve shows mice with the BACH1 gene, and the pink curve shows BACH1 deficient mice. The results, therefore, indicate that BACH1 contributes to longevity. Without the FGF21 gene, the presence or absence of BACH1 made no difference to lifespan. So, it was considered that the lifespan impact of BACH1 was due to FGF21 released upon ferroptosis. This research has brought new findings on the significance of ferroptosis. Cell death in some cells inhibits the aging of others, improves obesity and extends the lifespan. It was also suggested that BACH1, which promotes ferroptosis, is a gene for longevity. These results are still at the basic research stage, but if validated for application, it is expected to be applicable to the treatment of diseases associated with aging, including obesity and diabetes.